Hello everyone. This section is for growth curve. After learning this section, you should be able to describe the 4 phases of a microbial growth curve observed when microbes are grown in a batch culture. When microorganisms are cultivated in birth, they usually are grown in a batch culture. That is, they are incubated in a closed culture vessel with a single batch of media. Because no fresh media is provided during incubation, nutrient concentrations decline and concentrations of wastes increase. Population growth of microbes reproducing by binary fission in a batch culture can be plotted as a logarithm of the number of viable cells versus of the incubation time. The resulting curve has 4 distinct phases: lag phase, exponential phase, stationary phase, and death phase. Firstly, let's say lag phase. When microorganisms are introduced into fresh culture media, usually no immediate increase in cell number occurs. This period is called the lag phase. You can see the lag phase on the picture. It is not a time of inactivity. Rather, cells are synthesizing new components. There are many reasons for lag phase. This can be necessary for a variety of reasons. The cells may be old and depleted of ATP, essential cofactors, and ribosomes. These must be synthesized before growth can begin. The media may be different from the one the microorganisms were growing in previously. In this case, new enzymes are needed to use different nutrients. Possibly, the microorganisms have been injured and require time to recover. Eventually, however, the cells begin to replicate their DNA, increase in mass, and divide. As a result, the number of cells in the population begins to increase. The second phase is exponential phase. We also call it log phase. During this phase, microorganisms are growing and dividing at the maximal rate possible given their genetic potential, the nature of the media, and the environmental conditions. Their rate of growth is constant during the exponential phase. You can see the exponential phase on the picture. They are completing the cell cycle and doubling in number at regular intervals. The growth rate during log phase depends on several factors, including nutrient availability. When microbial growth is limited by the low concentration of a required nutrient, the final net growth or yield of cell increases with the initial amount of the limiting nutrient present. You can see it on Figure A. The rate of growth also increases with nutrient concentration on the Figure B, but a hyperbolic manner, much like that seen with many enzymes. The third phase is stationary phase. In a closed system, such as a batch culture, population growth eventually ceases, and the growth curve becomes horizontal. You can see the stationary phase on the picture. This stationary phase is attained by most bacteria at a population level of around 10 to the 9th cell per millimeter. In stationary phase, the total number of viable microorganisms remains constant. This may result from a balance between cell division and cell death, or the population may simply cease to divide, but remain metabolically active. One obvious reason microorganisms enter the stationary phase is nutrient limitation. If an essential nutrient is seriously depleted, population growth will slow and eventually stop. Microbes enter the stationary phase for other reasons besides nutrition limitation. Aerobic organisms often are limited by oxygen availability. Oxygen is not very soluble and may be depleted so quickly that only the surface of culture will have an oxygen concentration adequate for growth. Population growth also may cease due to the accumulation of toxic waste products. The last phase is death phase. Cell growing in batch culture cannot remain in stationary phase indefinitely. You can see the death phase on the picture. Eventually, they enter a phase that for many years was described simply as a death phase. During this phase, the number of viable cells often declines exponentially, with cells dying at a constant rate. It was assumed that detrimental environmental changes, such as nutrient deprivation and the buildup of toxic wastes, caused the irreparable harm to the cells. That is, even when bacterial cells were transferred to fresh media, no cellular growth was observed, because loss of viability was often not accompanied by a loss in total cell number. It was assumed that cells died, but did not lyse. At last, let's talk about mathematics of growth. Knowledge of microbial growth rates during the exponential phase is indispensable to microbiologists. Growth rate studies contribute to basic physiological and ecological research and are applied in industry. The quantitative aspects of exponential phase growth discussed here applied to microorganisms that divide by binary fission. During the exponential phase, each microorganism is dividing at constant intervals. Thus, the population doubles in number during a specific length of time. We call it generation or doubling time. This can be illustrated with a simple example. Suppose that a culture tube is inoculated with one cell that divides every 20 minutes. You can see it on the table. The population will be 2 cells after 20 minutes, 4 cells after 40 minutes, and so forth. Because the population is doubling every generation, the increase in population is always 2 to the nth, where n is the number of generations. The resulting population increase is exponential. Generation times vary markedly with the microbial species and environmental conditions. They range from less than 10 minutes to several days. You can see on the picture, we say E. coli. Under the incubation time, a temperature about 40 degrees, the generation time is 0.35 hours, and the mycobacteria tuberculosis under the temperature of 37. The generation time about 12 hours, and the Saccharomyces cerevisiae under the temperature of 30. The generation time is 2 hours. Generation times in nature are usually much longer than in culture. That's all for this section. Here is a word list for you.